Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Proteasome. 40 languages. Cartoon representation of a proteasome. Its active sites are sheltered inside the tube, blue. The caps, red, in this case, 11s. Regulatory particles, on the ends. Regulate entry into the destruction chamber, where the protein is degraded. Proteasomes are protein complexes which degrade ubiquitin. Tagged proteins by proteolysis, a chemical reaction that breaks peptide bonds. Enzymes that help such reactions are called proteases. Proteasomes are part of a major mechanism by which cells regulate the concentration of particular proteins and degrade misfolded proteins. Proteins are tagged for degradation with a small protein called ubiquitin. The tagging reaction is catalyzed by enzymes called ubiquitin ligases. Once a protein is tagged with a single ubiquitin molecule, this is a signal to other ligases to attach additional ubiquitin molecules. The result is a polyubiquitin chain that is bound by the proteasome, allowing it to degrade the tagged protein. 1. The degradation process yields peptides of about 7 to 8 amino acids long, which can then be further degraded into shorter amino acid sequences and used in synthesizing new proteins. 1. Proteasomes are found inside all eukaryotes and archaea, and in some bacteria. In eukaryotes, proteasomes are located both in the nucleus and in the cytoplasm. 2. In structure, the proteasome is a cylindrical complex containing a core of 4 stacked rings forming a central pore. Each ring is composed of 7 individual proteins. The inner 2 rings are made of 7 beta subunits that contain 3 to 7 protease active sites. These sites are located on the interior surface of the rings, so that the target protein must enter the central pore before it is degraded. The outer 2 rings each contain 7 alpha subunits whose function is to maintain a gate through which proteins enter the barrel. These alpha subunits are controlled by binding to cap. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Top view of the proteasome. Above. Structures or regulatory particles that recognize polyubiquitin. Tags attached to protein substrates and initiate the degradation process. The overall system of ubiquitination and proteasomal degradation is known as the ubiquitin proteasome system. 3. The proteasomal degradation pathway is essential for many cellular processes, including the cell cycle, the regulation of gene expression, and responses to oxidative stress. The importance of proteolytic degradation inside cells and the role of ubiquitin in proteolytic pathways was acknowledged in the award of the 2004 Nobel Prize in Chemistry to Aaron Sachanova, Avram Hershko and Erwin Rose. 4. Discovery, edit. Before the discovery of the ubiquitin proteasome system, protein degradation in cells was thought to rely mainly on lysosomes, membrane-bound organelles with acidic and protease-filled interiors that can degrade and then recycle exogenous proteins and aged or damaged organelles. 1. However, work by Joseph Ettlinger and Alfred L. Goldberg in 1977 on ATP-dependent protein degradation in reticulocytes, which lack lysosomes, suggested the presence of a second intracellular degradation mechanism. 5. This was shown in 1978 to be composed of several distinct protein chains, a novelty among proteases at the time. 6. Later work on modification of histones led to the identification of an unexpected covalent modification of the histone protein by a bond between a lysine side chain of the histone and the C-terminal glycine residue of ubiquitin, a protein that had no known function. 7. It was then discovered that a previously identified protein associated with proteolytic degradation, known as ATP-dependent proteolysis factor 1, APF1, was the same protein as ubiquitin. 8. The proteolytic activities of this system were isolated as a multiprotein complex originally called the multicatalytic proteinase complex by Sherwin Wilk and Marian Orlowski. 9. Later, the ATP-dependent proteolytic complex that was responsible for ubiquitin dependent protein degradation was discovered and was called the 26S proteasome. 10. 11. Much of the early work leading up to the discovery of the ubiquitin proteasome system occurred in the late 1970s and early 1980s at the Technion in the laboratory of Avram Hershko, where Aaron Sachanova worked as a graduate student. Hershko's year-long sabbatical in the laboratory of Erwin Rose at the Fox Chase Cancer Center provided key conceptual insights, though Rose later downplayed his role in the discovery. 12. The three shared the 2004 Nobel Prize in Chemistry for their work in discovering this system. 4. Although electron microscopy data revealing the stacked ring structure of the proteasome became available in the mid-1980s, 13. The first structure of the proteasome core particle was not solved by Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Schematic diagram of the Proteasome 20S core particle. Viewed from one side. The alpha subunits that make up the outer two rings are shown in green, and the beta subunits that make up the inner two rings are shown in blue. X-ray crystallography until 1994. 14. In 2018, the first atomic structures of the human 26S proteasome holoenzyme complex with a polyubiquitilated protein substrate were solved by cryogenic electron microscopy, revealing mechanisms by which the substrate is recognized, ubiquitilated, unfolded and degraded by the human 26S proteasome. 15. Structure and organization, edit. The proteasome subcomponents are often referred to by their Svedberg sedimentation coefficient, denoted S. The proteasome most exclusively used in mammals is the cytosolic 26S proteasome, which is about 2000 kilodaltons, K. The, in molecular mass containing 120S protein subunit and 219S regulatory cap subunits. The core is hollow and provides an enclosed cavity in which proteins are degraded. Openings at the two ends of the core allow the target protein to enter. Each end of the core particle associates with a 19S regulatory subunit that contains multiple ATPase active sites and ubiquitin binding sites. It is this structure that recognizes polyubiquitinated proteins and transfers them to the catalytic core. 15. An Alternative form of regulatory subunit called the 11s particle can associate with the core in essentially the same manner as the 19s particle. The 11s may play a role in degradation of foreign peptides such as those produced after infection by a virus. 16. 20s core particle, edit. 20s redirects here. For the decade, see 20s. The number and diversity of subunits contained in the 20s core particle depends on the organism. The number of distinct and specialized subunits is larger in multicellular than unicellular organisms, and larger in eukaryotes than in prokaryotes. All 20s particles consist of 4 stacked heptameric ring structures that are themselves composed of 2 different types of subunits. Alpha subunits are structural in nature, whereas beta subunits are predominantly catalytic. The alpha subunits are pseudoenzymes homologous to beta subunits. They are assembled with their N-termini adjacent to that of the beta subunits. 17. The outer 2 rings in the stack consist of 7 alpha subunits each, which serve as docking domains for the regulatory particles, and the alpha subunits N-termini, fam. PF 10584, form a gate that blocks unregulated access of substrates to the interior cavity. 18. The inner 2 rings each consist of 7 beta subunits and in their N-termini contain the protease active sites that perform the proteolysis reactions. 19. 3 distinct catalytic activities were identified in the purified complex, chymotrypsin-like, trypsin-like and peptidyl glutamyl peptide hydrolysing. 20. The size of the proteasome is relatively conserved and is about 150 angstroms, A, by 115 A. The Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Interior chamber is at most 53A wide, though the entrance can be as narrow as 13A, suggesting that substrate proteins must be at least partially unfolded to enter. 21. In archaea such as Thermoplasma acidophyllum, all the alpha and all the beta subunits are identical, whereas eukaryotic proteasomes such as those in yeast contain 7 distinct types of each subunit. In mammals, the 1, 2, and 5 subunits are catalytic, although they share a common mechanism. They have 3 distinct substrate specificities considered chymotrypsin-like, trypsin-like, and peptidyl glutamyl peptide hydrolysing, PHGH. 22. Alternative beta forms denoted 1I, 2I, and 5I can be expressed in hematopoietic cells in response to exposure to pro-inflammatory signals such as cytokines, in particular, interferon gamma. The proteasome assembled with these alternative subunits is known as the immunoproteasome, whose substrate specificity is altered relative to the normal proteasome. 21. Recently an alternative proteasome was identified in human cells that lack the 3 core subunit. 23. These proteasomes, known as the 4,4 proteasomes, instead form 20s core particles containing an additional 4 subunit in place of the missing 3 subunit. These alternative 4,4 proteasomes have been known previously to exist in yeast. 24. Although the precise function of these proteasome isoforms is still largely unknown, cells expressing these proteasomes show enhanced resistance to toxicity induced by metallic ions such as cadmium. 23, 25. 19s regulatory particle, edit. The 19S particle in eukaryotes consists of 19 individual proteins and is divisible into 2 subassemblies, a 9 subunit base that binds directly to the alpha ring of the 20S core particle, and a 10 subunit lid. 6 of the 9 base proteins are AT-parse subunits from the AAA family, and an evolutionary homologue of these AT-parses exists in archaea, called PAN, proteasome activating nucleotidase. 26. The association of the 19S and 20S particles requires the binding of ATP to the 19S AT-parse subunits, and ATP hydrolysis is required for the assembled complex to degrade folded and ubiquitinated proteins. Note that only the step of substrate unfolding requires energy from ATP hydrolysis, while ATP binding alone can support all the other steps required for protein degradation. For example, complex assembly, gate opening, translocation, and proteolysis. 27, 28. In fact, ATP binding to the AT-parses by itself supports the rapid degradation of unfolded proteins. However, while ATP hydrolysis is required for unfolding only, it is not yet clear whether this energy may be used in the coupling of some of these steps. 28, 29. In 2012, 2 independent efforts have elucidated the molecular architecture of the 26S proteasome by single particle electron microscopy. 31, 32. In 2016, 3 independent efforts have determined the first near-atomic resolution structure of the human 26S proteasome in the absence of substrates by cryo-EM. 33, 34, 35. In 2018, a major effort has elucidated the detailed mechanisms of dubiquitylation, initiation of translocation and processive unfolding of substrates by determining 7 atomic structures of substrate-engaged 26S proteasome simultaneously. 15. In the heart of explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF cartoon representation of the 26S proteasome. 30. The 19S, directly adjacent to the 20S, are the AAA apases. AAA proteins, that assemble to a heterohexameric ring of the order RPT1/RPT2/RPT6/RPT3/RPT4/RPT5. This ring is a trimer of dimers, RPT1/RPT2, RPT6/RPT3, and RPT4/RPT5 dimerize via their N-terminal coiled coils. These coiled coils protrude from the hexameric ring. The largest regulatory particle non-apases, RPN1 and RPN2, bind to the tips of RPT1/2 and RPT6/3, respectively. The ubiquitin receptor RPN13 binds to RPN2 and completes the base subcomplex. The lid covers 1/2 of the AAA apase hexamer, RPT6/RPT3/RPT4, and, unexpectedly, directly contacts the 20S via RPN6 and to lesser extent RPN5. The subunits RPN9, RPN5, RPN6, RPN7, RPN3, and RPN12, which are structurally related among themselves and to subunits of the COP9 complex and EIF3, hence called PCI. Subunits assemble to a horseshoe-like structure enclosing the RPN8/RPN11 heterodimer. RPN11, the ubiquitinating enzyme, is placed at the mouth of the AAA apase hexamer, ideally positioned to remove ubiquitin moieties immediately before translocation of substrates into the 20S. The second ubiquitin receptor identified to date, RPN10, is positioned at the periphery of the lid, near subunits RPN8 and RPN9. Conformational changes of 19S, edit. The 19S regulatory particle within the 26S proteasome holoenzyme has been observed in 6 strongly differing conformational states in the absence of substrates to date. 36, 37, a hallmark of the AAA apase configuration in this predominant low-energy state is a staircase or Lockwasher-like arrangement of the AAA domains. 30, 31, in the presence of ATP but absence of substrate 3. Alternative, less abundant conformations of the 19S are adopted, primarily differing in the positioning of the lid with respect to the AAA apase module. 33, 37, in the presence of ATPS or a substrate. Considerably more conformations have been observed displaying dramatic structural changes of the AAA apase module. 15, 36, 38, 39, some of the substrate-bound conformations bear high similarity to the substrate-free ones, but they are not entirely identical, particularly in the AAA AT-parse module. 15, 36, prior to the 26S assembly, the 19S regulatory particle in a free form has also been observed in 7 conformational states. 40, notably, all these conformers are somewhat different and present distinct features. Thus, the 19S regulatory particle can sample at least 20 conformational states under different physiological conditions. Regulation of the 20S by the 19S, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 3 distinct conformational states of the 26S proteasome. 37. The conformations are hypothesized to be responsible for recruitment of the substrate, its irreversible commitment, and finally processing and translocation into the core particle, where degradation occurs. The 19S regulatory particle is responsible for stimulating the 20S to degrade proteins. A primary function of the 19S regulatory AT-parses is to open the gate in the 20S that blocks the entry of substrates into the degradation chamber. 41. The mechanism by which the proteasomal AT-parse open this gate has been recently elucidated. 18. 20S gate opening, and thus substrate degradation, requires the C-termini of the proteasomal AT-parses, which contains a specific motif, i.e., HBYX motif. The AT-parses C-termini bind into pockets in the top of the 20S, and tether the AT-parse complex to the 20S proteolytic complex, thus joining the substrate unfolding equipment with the 20S degradation machinery. Binding of these C-termini into these 20S pockets by themselves stimulates opening of the gate in the 20S in much the same way that a key in a lock opens a door. 18. The precise mechanism by which this key in a lock mechanism functions has been structurally elucidated in the context of human 26S proteasome at near atomic resolution, suggesting that the insertion of 5 C-termini of AT parse subunits are PT1/2/3/5/6 into the 20s surface pockets are required to fully open the 20s gate. 36, 15, 33. Other regulatory particles, edit. 11s redirects here. Not to be confused with S11 or 11, plural. 20s proteasomes can also associate with a second type of regulatory particle, the 11s regulatory particle, a heptameric structure that does not contain any AT parses and can promote the degradation of short peptids but not of complete proteins. It is presumed that this is because the complex cannot unfold larger substrates. This structure is also known as PAR28, reg, or PAR26. 17. The mechanisms by which it binds to the core particle through the C-terminal tails of its subunits and induces ring conformational changes to open the 20s gate suggest a similar mechanism for the 19s particle. 42. The expression of the 11s particle is induced by interferon gamma and is responsible, in conjunction with the immunoproteasome beta subunits, for the generation of peptids that bind to the major histocompatibility complex. 16. Yet another type of non-apase regulatory particle is the BLM10, yeast, or PAR200/PSME4, human. It opens only one alpha subunit in the 20s gate and itself folds into a dome with a very small pore over it. 17. Assembly, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Ribbon diagram of ubiquitin, the highly conserved protein that serves as a molecular tag targeting proteins for degradation by the proteasome. The assembly of the proteasome is a complex process due to the number of subunits that must associate to form an active complex. The beta subunits are synthesized with N-terminal propeptids that are post-translationally modified during the assembly of the 20s particle to expose the proteolytic active site. The 20s particle is assembled from 2 half proteasomes, each of which consists of a 7-membered pro ring attached to a 7-membered alpha ring. The association of the beta rings of the 2 half proteasomes triggers 3-amine-dependent autolysis of the propeptids to expose the active site. These beta interactions are mediated mainly by salt bridges and hydrophobic interactions between conserved alpha helices whose disruption by mutation damages the proteasome's ability to assemble. 43. The assembly of the half proteasomes, in turn, is initiated by the assembly of the alpha subunits into their heptameric ring, forming a template for the association of the corresponding pro-ring. The assembly of alpha subunits has not been characterized. 44. Only recently, the assembly process of the 19s regulatory particle has been elucidated to considerable extent. The 19s regulatory particle assembles as 2 distinct subcomponents, the base and the lid. Assembly of the base complex is facilitated by 4 assembly chaperones. HSM3/S5B, NAS2/P27, RPN14/PAF1, and NAS6/gankirin, names for yeast/mammals. 45. These assembly chaperones bind to the AAA apase subunits and their main function seems to be to ensure proper assembly of the heterohexameric AAA apase ring. To date it is still under debate whether the base complex assembles separately, whether the assembly is templated by the 20S core particle, or whether alternative assembly pathways exist. In addition to the 4 assembly chaperones, the ubiquitinating enzyme UP6/USP14 also promotes base assembly, but it is not essential. 46. The lid assembles separately in a specific order and does not require assembly. Chaperones. 47. Protein degradation process, edit. Ubiquitination and targeting, edit. Further information, ubiquitination. Proteins are targeted for degradation by the proteasome, with covalent modification of a lysine residue that requires the coordinated reactions of 3 enzymes. In the first step, a ubiquitin activating enzyme, known as E1, hydrolyzes ATP and adenylylates a ubiquitin molecule. This is then transferred to E1's active site cysteine residue in concert with the adenylylation of a second ubiquitin. 48. This adenylylated ubiquitin is then transferred to a cysteine of a second enzyme, ubiquitin conjugating enzyme, E2. In the last step, a member of a highly diverse class of enzymes known as ubiquitin ligases, E3, recognizes the specific protein to be ubiquitinated and catalyzes the transfer of ubiquitin from E2 to. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The ubiquitination pathway. This target protein. A target protein must be labeled with at least 4 ubiquitin monomers, in the form of a polyubiquitin chain, before it is recognized by the proteasome lid. 49. It is therefore the E3 that confers substrate specificity to this system. 50. The number of E1, E2, and E3 proteins expressed depends on the organism and cell type, but there are many different E3 enzymes present in humans, indicating that there is a huge number of targets for the ubiquitin proteasome system. The mechanism by which a polyubiquitinated protein is targeted to the proteasome is not fully understood. A few high-resolution snapshots of the proteasome bound to a polyubiquitinated protein suggest that ubiquitin receptors might be coordinated with ubiquitinase RPN11 for initial substrate targeting and engagement. 15. Ubiquitin receptor proteins have an N-terminal ubiquitin-like, UBL, domain and one or more ubiquitin-associated, UBA, domains. The UBL domains are recognized by the 19S proteasome caps, and the UBA domains bind ubiquitin via 3 helix bundles. These receptor proteins may escort polyubiquitinated proteins to the proteasome, though the specifics of this interaction and its regulation are unclear. 51. The ubiquitin protein itself is 76 amino acids long and was named due to its ubiquitous nature, as it has a highly conserved sequence and is found in all known eukaryotic organisms. 52. The genes encoding ubiquitin in eukaryotes are arranged in tandem repeats, possibly due to the heavy transcription demands on these genes to produce enough ubiquitin for the cell. It has been proposed that ubiquitin is the slowest evolving protein identified to date. 53. Ubiquitin contains 7 lysine residues to which another ubiquitin can be ligated, resulting in different types of polyubiquitin chains. 54. Chains in which each additional ubiquitin is linked to lysine 48 of the previous ubiquitin have a role in proteasome targeting, while other types of chains may be involved in other Processes. 55, 56. Ubiquitination, edit. Ubiquitin chains conjugated to a protein targeted for proteasomal degradation are normally removed by any one of the three proteasome-associated ubiquitinating enzymes, DUBs, which are RPN11, UB6/USP14 and UCH37. This process recycles ubiquitin and is essential to maintain the ubiquitin reservoir in cells. 56. RPN11 is an intrinsic, stoichiometric subunit of the 19S regulatory particle and is essential for the function of 26S proteasome. The DUB activity of RPN11 is enhanced in the proteasome as compared to its monomeric form. How RPN11 removes a ubiquitin chain En block from a protein substrate was captured by an atomic structure of the substrate-engaged human proteasome in a conformation named B. 15. Interestingly, this structure also shows how the DUB activity is coupled to the substrate recognition by the proteasomal AAA apase. In contrast to explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. RPN11, USP14 and UCH37 are the DUBs that do not always associated with the proteasome. In cells, about 10 to 40% of the proteasomes were found to have USP14 associated. Both UB6/USP14 and UCH37 are largely activated by the proteasome and exhibit a very low DUB activity alone. Once activated, USP14 was found to suppress proteasome function by its DUB activity and by inducing parallel pathways of proteasome conformational transitions, one of which turned out to directly prohibit substrate insertion into the AAA apase, as intuitively observed by time-resolved cryogenic electron microscopy. 57. It appears that USP14 regulates proteasome function at multiple checkpoints by both catalytically competing with RPN11 and allosterically reprogramming the AAA 80 passe states, which is rather unexpected for a DUB. 57. These observations imply that the proteasome regulation may depend on its dynamic transitions of conformational states. Unfolding and translocation, edit. After a protein has been ubiquitinated, it is recognized by the 19S regulatory particle in an ATP-dependent binding step. 15. 28. The substrate protein must then enter the interior of the 20S subunit to come in contact with the proteolytic active sites. Because the 20S particle's central channel is narrow and gated by the N-terminal tails of the alpha ring subunits, the substrates must be at least partially unfolded before they enter the core. 15. The passage of the unfolded substrate into the core is called translocation and necessarily occurs after ubiquitination. 15. 28. However, the order in which substrates are ubiquitinated and unfolded is not yet clear. 58. Which of these processes is the rate-limiting step in the overall proteolysis reaction depends on the specific substrate. For some proteins, the unfolding process is rate-limiting, while ubiquitination is the slowest step for other proteins. 27. The extent to which substrates must be unfolded before translocation is suggested to be around 20 amino acid residues by the atomic structure of the substrate-engaged 26S proteasome in the ubiquitination compatible state. 15. But substantial tertiary structure, and in particular non-local interactions such as disulfide bonds, are sufficient to inhibit degradation. 59. The presence of intrinsically disordered protein segments of sufficient size, either at the protein terminus or internally, has also been proposed to facilitate efficient initiation of degradation. 61. The gate formed by the alpha subunits prevents peptides longer than about 4 residues from entering the interior of the 20S particle. The ATP molecules bound before the initial recognition step are hydrolysed before translocation. While energy is needed for substrate unfolding, it is not required for translocation. 28. The assembled 26S proteasome can degrade unfolded proteins in the presence of a non-hydrolysable ATP analog, but cannot degrade folded proteins, indicating that energy from ATP hydrolysis is used for substrate unfolding. 27. Passage of the unfolded substrate through the opened gate occurs via facilitated diffusion if the 19S cap is in the ATP-bound state. 62. The mechanism for unfolding of globular proteins is necessarily general, but somewhat dependent on the amino acid sequence. Long sequences of alternating glycine and alanine have been shown to inhibit substrate unfolding, decreasing the efficiency of proteasomal degradation. This results in explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. A cutaway view of the Proteasome 20S core particle, illustrating the locations of the active sites. The alpha subunits are represented as green spheres, and the beta subunits as protein backbones colored by individual polypeptide chain. The small pink spheres represent the location of the active site threonine residue in each subunit. Light blue chemical structures are the inhibitor bortezomib bound to the active sites. The release of partially degraded byproducts, possibly due to the decoupling of the ATP hydrolysis and unfolding steps. 63. Such glycine-alanine repeats are also found in nature, for example in silk fibroin. In particular, certain Epstein-Barr virus gene products bearing this sequence can stall the proteasome, helping the virus propagate by preventing antigen presentation on the major histocompatibility complex. 64. Proteolysis, edit. See also, threonine protease mechanism. The proteasome functions as an endoprotease. 65, 66, 67, 68. The mechanism of proteolysis by the beta subunits of the 20S core particle is through a threonine-dependent nucleophilic attack. This mechanism may depend on an associated water molecule for deprotonation of the reactive threonine hydroxyl. Degradation occurs within the central chamber formed by the association of the two beta rings and normally does not release partially degraded products, instead reducing the substrate to short polypeptides, typically 7-9 residues long, though they can range from 4 to 25 residues, depending on the organism and substrate. The biochemical mechanism that determines product length is not fully characterized. 69. Although the three catalytic beta subunits have a common mechanism, they have slightly different substrate specificities, which are considered chymotrypsin-like, trypsin-like, and peptidyl glutamyl peptide hydrolysing, PHGH, like. These Variations in specificity are the result of interatomic contacts with local residues near the active sites of each subunit. Each catalytic beta subunit also possesses a conserved lysine residue required for proteolysis. 22. Although the proteasome normally produces very short peptide fragments, in some cases these products are themselves biologically active and functional molecules. Certain transcription factors regulating the expression of specific genes, including one component of the mammalian complex NFB, are synthesized as inactive precursors whose ubiquitination and subsequent proteasomal degradation converts them to an active form. Such activity requires the proteasome to cleave the substrate protein internally, rather than processively degrading it from one terminus. It has been suggested that long loops on these protein surfaces serve as the proteasomal substrates and enter the central cavity, while the majority of the protein remains outside. 70. Similar effects have been observed in yeast proteins. This mechanism of selective degradation is known as regulated ubiquitin /proteasome dependent processing, RUP. 71. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The assembled complex of HSLV, blue, and HSLU, red, from E. coli. This complex of heat shock proteins is thought to resemble the ancestor of the modern proteasome. Ubiquitin-independent degradation, edit. Although most proteasomal substrates must be ubiquitinated before being degraded, there are some exceptions to this general rule, especially when the proteasome plays a normal role in the post-translational processing of the protein. The proteasomal activation of NFB by processing P105 into 50P via internal proteolysis is one major example. 70. Some proteins that are hypothesized to be unstable due to intrinsically unstructured regions, 72, are degraded in a ubiquitin-independent manner. The most well-known example of a ubiquitin-independent proteasome substrate is the enzyme ornithine decarboxylase. 73. Ubiquitin-independent mechanisms targeting key cell cycle regulators such as 53P have also been reported, although 53P is also subject to ubiquitin-dependent degradation. 74. Finally, structurally abnormal, misfolded, or highly oxidized proteins are also subject to ubiquitin-independent and 19S-independent degradation under conditions of cellular stress. 75. Evolution, edit. The 20S proteasome is both ubiquitous and essential in eukaryotes and archaea. The bacterial order Actinomysteales also share homologs of the 20S proteasome, whereas most bacteria possess heat shock genes HSLV and HSLU, whose gene products are a multimeric protease arranged in a two-layered ring and an AT-parsec. 76. The HSLV protein has been hypothesized to resemble the likely ancestor of the 20S proteasome. 77. In general, HSLV is not essential in bacteria, and not all bacteria possess it, whereas some protists possess both the 20S and the HSLV systems. 76. Many bacteria also possess other homologs of the proteasome and an associated AT-parsec, most notably CLPP and CLPX. This redundancy explains why the HSLUV system is not essential. Sequence analysis suggests that the catalytic beta subunits diverged earlier in evolution than the predominantly structural alpha subunits. In bacteria that express a 20S proteasome, the beta subunits have high sequence identity to archaeal and eukaryotic beta subunits, whereas the alpha sequence identity is much lower. The presence of 20S proteasomes in bacteria may result from lateral gene transfer, while the diversification of subunits among eukaryotes is ascribed to multiple gene duplication events. 76. Cell cycle control, edit. Cell cycle progression is controlled by ordered action of cyclin-dependent kinases, CDKs, activated by specific cyclins that demarcate phases of the cell cycle. Mitotic cyclins, which persist in. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The cell for only a few minutes, have one of the shortest lifespans of all intracellular proteins. 1. After a CDK cyclin complex has performed its function, the associated cyclin is polyubiquitinated and destroyed by the proteasome, which provides directionality for the cell cycle. In particular, exit from mitosis requires the proteasome-dependent dissociation of the regulatory component cyclin B from the mitosis-promoting factor complex. 78. In vertebrate cells, slippage through the mitotic checkpoint leading to premature M-phase exit can occur despite the delay of this exit by the spindle checkpoint. 79. Earlier cell cycle checkpoints such as post-restriction point check between G1 phase and S phase similarly involve proteasomal degradation of cyclin A, whose ubiquitination is promoted by the anaphase promoting complex, APC, an E3 ubiquitin ligase. 80. The APC and the SKP1/Cul1/F box protein complex, SCF complex, are the two key regulators of cyclin degradation and checkpoint control. The SCF itself is regulated by the APC via ubiquitination of the adapter protein, SKP2, which prevents SCF activity before the G1S transition. 81. Individual components of the 19S particle have their own regulatory roles. Gankirin, a recently identified oncoprotein, is one of the 19S subcomponents that also tightly binds the cyclin-dependent kinase CDK4 and plays a key role in recognizing ubiquitinated 53P, via its affinity for the ubiquitin ligase MAM2. Gankirin is anti-apoptotic and has been shown to be overexpressed in some tumor cell types such as hepatocellular carcinoma. 82. Like eukaryotes, some archaea also use the proteasome to control cell cycle, specifically by controlling ISCT3-mediated cell division. 83. Regulation of plant growth, edit. In plants, signaling by auxins, or phytohormones that order the direction and tropism of plant growth, induces the targeting of a class of transcription factor repressors known as auxiliary/IAA proteins for proteasomal degradation. These proteins are ubiquitinated by SCF1, or SCF in complex with the auxin receptor TIR1. Degradation of auxiliary/IAA proteins depresses transcription factors in the auxin response factor, ARF, family and induces ARF-directed gene expression. 84. The cellular consequences of ARF activation depend on the plant type and developmental stage, but are involved in directing growth in roots and leaf veins. The specific response to ARF depression is thought to be mediated by specificity in the pairing of individual ARF and auxiliary /IAA proteins. 85. Apoptosis, edit. Both internal and external signals can lead to the induction of apoptosis, or programmed cell death. The resulting deconstruction of cellular components is primarily carried out by specialized proteases known as caspases, but the proteasome also plays important and diverse roles in the apoptotic process. The involvement of the proteasome in this process is indicated by both the Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Increase in protein ubiquitination, and of E1, E2, and E3 enzymes that is observed well in advance of apoptosis. 86, 87, 88. During apoptosis, proteasomes localized to the nucleus have also been observed to translocate to outer membrane blebs characteristic of apoptosis. 89. Proteasome inhibition has different effects on apoptosis induction in different cell types. In general, the proteasome is not required for apoptosis, although inhibiting it is pro-apoptotic in most cell types that have been studied. Apoptosis is mediated through disrupting the regulated degradation of pro-growth cell cycle proteins. 90. However, some cell lines, in particular, primary cultures of quiescent and differentiated cells such as thymocytes and neurons, are prevented from undergoing apoptosis on exposure to proteasome inhibitors. The mechanism for this effect is not clear, but is hypothesized to be specific to cells in quiescent states, or to result from the differential activity of the pro-apoptotic kinase JNK. 91. The ability of proteasome inhibitors to induce apoptosis in rapidly dividing cells has been exploited in several recently developed chemotherapy agents such as bortezomib and selinospiramide. Response to cellular stress, edit. In response to cellular stresses such as infection, heat shock, or oxidative damage heat shock, proteins that identify misfolded or unfolded proteins and target them for proteasomal degradation are expressed. Both HSP27 and HSP90, chaperone proteins, have been implicated in increasing the activity of the ubiquitin proteasome system, though they are not direct participants in the process. 92. HSP70, on the other hand, binds exposed hydrophobic patches on the surface of misfolded proteins and recruits E3 ubiquitin ligases such as CHIP to tag the proteins for proteasomal degradation. 93. The CHIP protein, carboxyl terminus of HSP70 interacting protein, is itself regulated via inhibition of interactions between the E3 enzyme CHIP and its E2 binding partner. 94. Similar mechanisms exist to promote the degradation of oxidatively damaged proteins via the proteasome system. In particular, proteasomes localized to the nucleus are regulated by PARP and actively degrade inappropriately oxidized histones. 95. Oxidized proteins, which often form large amorphous aggregates in the cell, can be degraded directly by the 20S core particle without the 19S regulatory cap and do not require ATP hydrolysis or tagging with ubiquitin. 75. However, high levels of oxidative damage increases the degree of cross-linking between protein fragments, rendering the aggregates resistant to proteolysis. Larger numbers and sizes of such highly oxidized aggregates are associated with aging. 96. Dysregulation of the ubiquitin proteasome system may contribute to several neural diseases. It may lead to brain tumors such as astrocytoma. 97. In some of the late-onset neurodegenerative diseases that share aggregation of misfolded proteins as a common feature, such as Parkinson's disease and Alzheimer's disease, large insoluble aggregates of misfolded proteins can form and explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Then result in neurotoxicity, through mechanisms that are not yet well understood. Decreased proteasome activity has been suggested as a cause of aggregation and Lewy body formation in Parkinson's. 98. This hypothesis is supported by the observation that yeast models of Parkinson's are more susceptible to toxicity from synuclein, the major protein component of Lewy bodies, under conditions of low proteasome activity. 99. Impaired proteasomal activity may underlie cognitive disorders such as the autism spectrum disorders, and muscle and nerve diseases such as inclusion body myopathy. 97. Role in the immune system, edit. The proteasome plays a straightforward but critical role in the function of the adaptive immune system. Peptide antigens are displayed by the major histocompatibility complex class I, MHC, proteins on the surface of antigen-presenting cells. These peptides are products of proteasomal degradation of proteins originated by the invading pathogen. Although constitutively expressed, proteasomes can participate in this process, a specialized complex composed of proteins, whose expression is induced by interferon gamma, are the primary producers of peptides which are optimal in size and composition for MHC binding. These proteins whose expression increases during the immune response include the 11S regulatory particle, whose main known biological role is regulating the production of MHC ligands, and specialized beta subunits called 1I, 2I, and 5I with altered substrate specificity. The complex formed with the specialized beta subunits is known as the immunoproteasome. 16. Another 5I variant subunit, 5T, is expressed in the thymus, leading to a thymus-specific thymoproteasome whose function is as yet unclear. 100. The strength of MHC class I ligand binding is dependent on the composition of the ligand C terminus, as peptides bind by hydrogen bonding and by close contacts with a region called the B pocket on the MHC surface. Many MHC class I alleles prefer hydrophobic C-terminal residues, and the immunoproteasome complex is more likely to generate hydrophobic C-termini. 101. Due to its role in generating the activated form of NFB, an anti-apoptotic and pro-inflammatory regulator of cytokine expression, proteasomal activity has been linked to inflammatory and autoimmune diseases. Increased levels of proteasome activity correlate with disease activity and have been implicated in autoimmune diseases including systemic lupus erythematosus and rheumatoid arthritis. 16. The proteasome is also involved in intracellular antibody-mediated proteolysis of antibody-bound virions. In this neutralization pathway, TRIM21, a protein of the tripartite motif family, binds with immunoglobulin G to direct the virion to the proteasome where it is degraded. 102. Proteasome inhibitors, edit. Main article, Proteasome inhibitor. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Chemical structure of bortezomib, baronated form of Mg132, a proteasome inhibitor used in chemotherapy that is particularly effective against multiple myeloma. Bortezomib bound to the core particle in a yeast proteasome. The bortezomib molecule is in the center, colored by atom type: carbon equals pink, nitrogen equals blue, oxygen equals red, boron equals yellow. Surrounded by the local protein surface. The blue patch is the catalytic threonine residue, whose activity is blocked by the presence of bortezomib. Proteasome inhibitors have effective antitumor activity in cell culture, inducing apoptosis by disrupting the regulated degradation of pro-growth cell cycle proteins. 90. This approach of selectively inducing apoptosis in tumor cells has proven effective in animal models and human trials. Lactacystin, a natural product synthesized by Streptomyces bacteria, was the first non-peptidic proteasome inhibitor discovered. 103, and is widely used as a research tool in biochemistry and cell biology. Lactacystin was licensed to Myogenics/Prescript, which was acquired by Millennium Pharmaceuticals, now part of Takeda Pharmaceuticals. Lactacystin covalently modifies the amino-terminal threonine of catalytic beta subunits of the proteasome, particularly the 5 subunit responsible for the proteasome's chymotrypsin-like activity. This discovery helped to establish the proteasome as a mechanistically novel class of protease, an amino-terminal threonine protease. Bortezomib, baronated Mg132, a molecule developed by Millennium Pharmaceuticals and marketed as Velcade, is the first proteasome inhibitor to reach clinical use as a chemotherapy agent. 104. Bortezomib is used in the treatment of multiple myeloma. 105. Notably, multiple myeloma has been observed to result in increased proteasome-derived peptide levels in blood serum that decrease to normal levels in response to successful chemotherapy. 106. Studies in animals have indicated that bortezomib may also have clinically significant effects in pancreatic cancer. 107, 108. Preclinical and early clinical studies have been started to examine bortezomib's effectiveness in treating other B-cell-related cancers. 109, particularly some types of non-Hodgkin's lymphoma. 110. Clinical results also seem to justify use of proteasome inhibitor combined with chemotherapy for B-cell acute lymphoblastic leukemia. 111. Proteasome inhibitors can kill some types of cultured leukemia cells that are resistant to glucocorticoids. 112. The molecule ritonavir, marketed as Norfa, was developed as a protease inhibitor and used to target HIV infection. However, it has been shown to inhibit proteasomes as well as free proteases. To be specific, the chymotrypsin-like activity of the proteasome is inhibited by ritonavir, while the trypsin-like activity is somewhat enhanced. 113. Studies in animal models suggest that ritonavir may have inhibitory effects on the growth of glioma cells. 114. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Proteasome inhibitors have also shown promise in treating autoimmune diseases in animal models. For example, studies in mice-bearing human skin grafts found a reduction in the size of lesions from psoriasis after treatment with a proteasome inhibitor. 115. Inhibitors also show positive effects in rodent models of asthma. 116. Labeling and inhibition of the proteasome is also of interest in laboratory settings for both in vitro and in vivo study of proteasomal activity in cells. The most commonly used laboratory inhibitors are lactacystin and the peptidaldehyde MG132, initially developed by Goldberg Lab. Fluorescent inhibitors have also been developed to specifically label the active sites of the assembled proteasome. 117. Clinical significance, edit. The proteasome and its subunits are of clinical significance for at least two reasons. 1. A compromised complex assembly or a dysfunctional proteasome can be associated with the underlying pathophysiology of specific diseases, and 2. They can be exploited as drug targets for therapeutic interventions. More recently, more effort has been made to consider the proteasome for the development of novel diagnostic markers and strategies. An improved and comprehensive understanding of the pathophysiology of the proteasome should lead to clinical applications in the future. The proteasomes form a pivotal component for the ubiquitin proteasome system, UPS 118, and corresponding cellular protein quality control, PQC. Protein ubiquitination and subsequent proteolysis and degradation by the proteasome are important mechanisms in the regulation of the cell cycle, cell growth and differentiation, gene transcription, signal transduction and apoptosis. 119. Proteasome defects lead to reduced proteolytic activity and the accumulation of damaged or misfolded proteins, which may contribute to neurodegenerative disease. 120, 121, cardiovascular diseases, 122, 123, 124, inflammatory responses and autoimmune diseases, 125, and systemic DNA damage responses leading to malignancies. 126. Research has implicated UPS defects in the pathogenesis of neurodegenerative and myodegenerative disorders, including Alzheimer's disease, 127, Parkinson's disease, 128, and Pick's disease, 129, amyotrophic lateral sclerosis, ALS, 129, Huntington's disease, 128, Creutzfeldt-Jakob disease, 130, and motor neuron diseases, polyglutamine, polyQ diseases, muscular dystrophies, 131, and several rare forms of neurodegenerative diseases associated with dementia. 132, as part of the ubiquitin proteasome system, UPS, the proteasome maintains cardiac protein homeostasis and thus plays a significant role in cardiac ischemic injury. 133. Ventricular hypertrophy, 134, and heart failure. 135. Additionally, evidence is accumulating that the UPS plays an essential role in malignant transformation. UPS proteolysis plays a major role in responses of cancer cells to stimulatory signals that are critical for the development of cancer. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Accordingly, gene expression by degradation of transcription factors, such as 53p, cJUN, cFUS, NF, B, cMyc, HIF1, MAT2, STAT3, sterol regulated element binding proteins and androgen receptors are all controlled by the UPS and thus involved in the development of various malignancies. 136. Moreover, the UPS regulates the degradation of tumor suppressor gene products, such as adenomatous polyposis coli, APC, in colorectal cancer, retinoblastoma, RB, and von Hippel-Lindau tumor suppressor, VHL, as well as a number of proto-oncogenes, RAF, MYC, MYB, RIL, SRC, MOS, ABL. The UPS is also involved in the regulation of inflammatory responses. This activity is usually attributed to the role of proteasomes in the activation of NFB, which further regulates the expression of pro-inflammatory cytokines such as TNF, IL, IL-8, adhesion molecules, ICAM-1, VCAM-1, P-selectin, and prostaglandins and nitric oxide, NO. 125. Additionally, the UPS also plays a role in inflammatory responses as regulators of leukocyte proliferation, mainly through proteolysis of cyclines and the degradation of CDK inhibitors. 137. Lastly, autoimmune disease patients with SLE, Sjogren syndrome and rheumatoid arthritis, RA, predominantly exhibit circulating proteasomes which can be applied as clinical biomarkers. 138. See also, edit the proteolysis map. DSS1/SEM1 protein family. Exosome complex. Endoplasmic reticulum associated protein degradation. Junk and iPod. References, edit. 1 ABCD Lodish H, Burko, Matsudera P, Kaiser CA, Krieger M, Scott MP, Zipersky SL, Darnell J, 2004. 3. Molecular Cell Biology, 5th ed. New York, W.H. Freeman & Co. pp. 66-72. ISBN 978-0. 7167-4366-8. 2 Peters J.M., Frank W.W., Kleinschmidt J.A., March 1994. Distinct 19S and 20S subcomplexes of the 26S proteasome and their distribution in the nucleus and the cytoplasm. The Journal of Biological Chemistry. 269, 107709 18 do. 10.1016/S0021-9258, 17, 37345-3. PMID 8125997. 3 Narsif, Nicholas D. Cambrai, Samantha E. Kraut, Daniela. May 2014. Slipping up, partial substrate degradation by ATP-dependent proteases. EUBM Life. 66, 5309317. Do. 10.1002/EUB.1271. PMID 248239773. S2 SID 2986028. 4 AB Nobel Prize Committee, 2004. Nobel Prize awardees in Chemistry, 2004. Retrieved. the 11th of December, 2006. 5 Etterlinger J.D., Goldberg al., January 1977. A soluble ATP-dependent proteolytic system responsible for the degradation of abnormal proteins in reticulocytes. Proceedings of the National Academy of Sciences of the United States of America. 74, 154-8. Bibcode, 1977 NAS, 74, 54E. Do. 10.1073/NAS.74.1.54. PMC 393195. PMID 264694. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 6 Sihanover, Hodwy, Hershkower, April 1978. A heat-stable polypeptide component of an ATP dependent proteolytic system from reticulocytes. Biochemical and Biophysical Research. Communications. 81, 41105 to 10.1016/0006-291X, 78, 91249-4. PMID 666810. 7 Goldknopf L., Busch, March 1977. Isopeptide linkage between nonhistone and histone 2AM. Polypeptides of chromosomal conjugate protein A24. Proceedings of the National Academy of Sciences of the United States of America. 74, 38648. Bibcode, 1977 NAS, 74864G. Do. 10.1073/NAS.74.3.864. PMC 430507. PMID 265581. 8 Sichanova, September 2005. Early work on the ubiquitin proteasome system, an interview with Aaron Sichanova. Interview by CDD. Cell death and differentiation. 12, 91167-77. Do. 10.1038/sj.cdd.4401691. PMID 16094393. 9 Wilk S., Orlowski M., November 1980. Cation-sensitive neutral endopeptidase, isolation and specificity of the bovine pituitary enzyme. Journal of Neurochemistry. 35, 51172-82. Do. 10.101411-4171-4159.1980.TB07873.X. PMID 6778972. S2 SID 9028201. 10 Irigo AP, Tanaka, K., Goldberg F., Welch W.J., 1988. Identity of 19S prosome particle with the large multifunctional protease complex of mammalian cells. Nature. 331, 6152-19294. Do. 10.1038/3311920. PMID 3277060. S2 SID 97688. Tanaka K., Waxman L., Goldberg L., June 1983. ATP serves two distinct roles in protein degradation in reticulocytes, one requiring and one Independent of ubiquitin. The Journal of Cell Biology. 96, 1585 do. 10.1083/jcb.96.6.1580. PMC 2112434. PMID 6304111. 11 Huffer, Pratt G., Reeksteiner M., June 1987. Purification of 2 high molecular weight proteases. From rabbit reticulocyte lysate. The Journal of Biological Chemistry. 262, 18303 do. 10.1016/S0021-9258, 18, 47564-3. PMID 3298229. 12 Hershkower, September 2005. Early work on the ubiquitin proteasome system, an interview with Avram Hershko. Interview by CDD. Cell death and differentiation. 12, 9 1158-61. do. 10.1038/SJ.CDD.4401709. PMID 16094391. 13 Cop F, Steiner R, Dahlman B, Kuhnel, Reinauer H, August 1986. Size and shape of the multicatalytic proteinase from rat skeletal muscle. Biochimica et Biophysica Acta, BBA, protein. Structure and molecular enzymology. 872, 325362, 10.1016/0167-4838, 86, 90278-5. PMID 35246888. 14 Low J, Stock D, Jap B, Zwicky P, Baumeister W, Huber R, April 1995. Crystal structure of the 20S. Proteasome from the archaeon T acidophilum at 3.4 resolution. Science. 268, 5210-5339. Bibcode 1995 SCI 268533L. do. 10.1111/science.77250097. PMID 77250097. 15 ABCDEFGHIJKL Dong Y, Zhang S, Wu Z, Li X, Wang W L, Chu Y, Stoilova McPhee S, Lu Y, Finley D. Mao Y, November 2018. Cryo-M Structures and Dynamics of Substrate-Engaged Human 26S. Proteasome. Nature. 565, 7737 4955 do. 10.1038/S41586-018-07364. PMC 6370054. PMID 30479383. 16 ABCD Wang J, Maldonado Ma, August 2006. The ubiquitin proteasome system and its role in inflammatory and autoimmune diseases. Cellular and molecular immunology. 3, 425561. PMID 16978533. 17 ABC Stadtmüller, B.M., Hill, C.P., the 7th of January 2011. Proteasome activators. Molecular cell. 41, 1. 819. do. 10.1016/j.mulchel.2010.12.020. PMC 3040445. PMID 21211719. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 18-carat ABC Smith DM, Chang SC, Park S, Finley D, Cheng Y, Goldberg L, September 2007. Docking of the proteasomal 80 parses carboxyl termini in the 20s proteasomes alpha ring opens the gate for substrate entry. Molecular cell 27, 573144 do 10.1016/j.mulchel.2007.06.033. PMC 2083707. PMID 17803938. 19-carat Merops family T1. EMBLEBI. Retrieved 16 February 2019. 20-carat Wilk S, Orlowski M, March 1983. Evidence that pituitary cation-sensitive neutral endopeptidase is a multicatalytic protease complex. Journal of Neurochemistry. 40, 38429 do. 10.1111/j.1471. 4159.1983.tb08056.x. PMID 63388156. S2 SID 2350865. 21-carat AB Nandi D, Tahiliani P, Kumara, Chandu D, March 2006. The Ubiquitin Proteasome System. PDF. Journal of Biosciences. 31, 113755 do. 10.1007/BF02705243. PMID 165958883. S2 SID 21603835. 22-carat AB Heinemeyer W, Fisher M, Krumer T, Starch on U, Wolf DH, October 1997. The active sites of the eukaryotic 20s proteasome and their involvement in subunit precursor processing. The Journal of Biological Chemistry. 272, 42520009 do. 10.1074/jbc.272.40.25200. PMID 93121334. 23-carat AB Padmanabhorn, Vuong Sat, Hochstrasser M, March 2016. Assembly of an evolutionarily conserved alternative proteasome isoform in human cells. Cell Reports. 14, 12 2962-74. do. 10.1016/j.cellrip.2016.02.068. PMC 48287229. PMID 269972668. 24-carat Velishutina I, Connolly P.L., Arendt C.S., Lee X., Hochstrasser M., February 2004. Plasticity in eukaryotic 20s proteasome ring assembly revealed by a subunit deletion in yeast. The Embo Journal. 23, 3. 510. do. 10.1038/sj.embodge.76000059. PMC 1271798. PMID 147399934. 25-carat Kusmi Urchika, Kunjapu MJ, Funakoshi M., Hochstrasse M., March 2008. A multimeric assembly factor controls the formation of alternative 20S proteasomes. Nature, Structural and Molecular Biology. 15. 323744 do. 10.1038/NSMB.1389. PMID 18278055. S2 SID 21181637. 26 carats wiki ing d. Kilometer, Klenk HP, Goldberg L. September 1999. An archaebacterial AT parse. Homologous to AT parses in the eukaryotic 26S proteasome, activates protein breakdown by 20S. Proteasomes. The Journal of Biological Chemistry. 274, 3726000814. Do. 10.1074/JBC.274.37.26008. PMID 10473546. 27 carat ABCD Smith DM, Cafrigi, Cheng Y, Ing D, Was T, Goldberg L, December 2005. ATP binding to PAN or the 26S AT parses causes association with the 20S proteasome, gate opening, and translocation of unfolded proteins. Molecular cell. 20, 568798 do. 10.1016/J.Molchel.2005.10.019. PMID 163375993. 28 carat ABCDE Lucy W, Lee X, Thomson D, Wooding K, Chang T L, Tang Z, UH, Thomas P J, DeMartino G N. October 2006. ATP binding and ATP hydrolysis play distinct roles in the function of 26S. Proteasome. Molecular cell. 24, 13950 do. 10.1016/J.Molchel.2006.08.025. PMC 395117. PMID 17018291. 29 carat Lamia, Lawson TG, Valeutum M, Zweier JL, Picard cm, April 2002. A proteasomal AT parse. Subunit recognizes the polyubiquitin degradation signal. Nature. 416, 6882 7637. Bibcode 2002 Nature.4167663L. Do. 10.1038/4167638. PMID 11961560. S2 SID 44211764. 30 carat AB Beck F, Unverdor Ben P, Bonn S, Schweitzer, Pfeffer G, Sakater E, Nickel S, Plitzko JM, Villa E. Baumeister W, Forster F, September 2012. Near atomic resolution structural model of the yeast 26S. Proteasome. Proceedings of the National Academy of Sciences of the United States of America. 109. 37148705. Bibcode 2012 NAS 109148770B. Do. 10.1073/NAS.12133333109. PMC 34431224. PMID 229273775. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 31 AB Landa GC, Estrini, Matisculami, Bashor C, Nogale Z, Martino, February 2012. Complete. Subunit Architecture of the Proteasome Regulatory Particle. Nature. 482, 7384 18691. Bibcode, 2012 Nature.482186L. Do. 10.1038/Nature10774. PMC 3285539. PMID 222370024. 32 Lasker K, Forster F, Bon S, Walls Duaniti, Villa E, Unverdor Ben P, Beck F, Ibersold R, Celio. Baumeister W, January 2012. Molecular architecture of the 26S proteasome holocomplex determined by an integrative approach. Proceedings of the National Academy of Sciences of the United States of America. 109, 1387 do. 10.1073/NAS.11205599109. PMC 3277140. PMID 223075889. 33 ABC Chen S, Wu J, Lu Y, Ma YB, Li BH, UZ, Yang Q, Finley DJ, Kirschner MW, Mao Y. November 2016. Structural basis for dynamic regulation of the human 26S proteasome. Proceedings of the National Academy of Sciences of the United States of America. 113, 46 12991. 12996 Bibcode, 2016 NAS 113129991C. Do. 10.1073/NAS.16146141113. PMC 5135334. PMID 277911164. 34 Huang X, Luan B, Wu J, Shi Y, September 2016. Anatomic structure of the human 26S proteasome. Nature Structural and Molecular Biology. 23, 9778785 do. 10.1038/NSMB.3273. PMID 274287775. S2 SID 219093333. 35 Schweitzer, Aufderheide, Rudakti, Beck F, Pfeffer G, Plitzko JM, Sakater E, Skulton K, Forster F, Baumeister W, July 2016. Structure of the human 26S proteasome at a resolution of 3.9a. Proceedings of the National Academy of Sciences of the United States of America. 113, 28 7816. 7821 Bibcode, 2016 NAS 113129991C. Do. 10.1073/NAS.16146141113. PMC 5135334. PMID 277911164. 36 ABCDGUY, Wang WL, UD, Yang Q, Lu Y, Mao Y, April 2018. Structural mechanism for nucleotide-driven remodeling of the AAA apase unfoldase in the activated human 26S proteasome. Nature Communications. 9, 1360. Bibcode, 2018 Natco, 9.1360Z. Do 10.1038/S41467-018-037885W. PMC 5893597. PMID 2963647-2. 37 ABC Unverdorben P. Beck F. Sleds P. Schweitzer, Pfeffer G. Plitzko J.M. Baumeister W. Forster F. April 2014. Deep classification of a large Creo-M dataset defines the conformational landscape of the 26S proteasome. Proceedings of the National Academy of Sciences of the United States of America. 111, 15 5544 9. Bib code 2014 as 111.5544 U. Do 10.1073/NAS.14034091111. PMC 3992697. PMID 247068444. 38 Sleds P. Unverdorben P. Beck F. Pfeffer G. Schweitzer, Forster F. Baumeister W. April 2013. Structure of the 26S proteasome with ATPase bound provides insights into the mechanism of nucleotide-dependent substrate translocation. Proceedings of the National Academy of Sciences of the United States of America. 110, 18 7264 7269. Bib code 2013 as 110.7264S. Do 10.1073/NAS.13057821110. PMC 3645540. PMID 235898442. 39 Matiscalemi, Landa G.C., Martino, July 2013. Conformational switching of the 26S proteasome enables substrate degradation. Nature Structural and Molecular Biology. 20, 77817888. Do 10.1038/NSMB.2616. PMC 3712289. PMID 237708119. 40 Lu Y, Wu J, Dong Y, Chen S, Sun S, Ma YB, Yang Q, Finley D, Kirschner MW, Mao Y, July 2017. Conformational landscape of the P28-bound human proteasome regulatory particle. Molecular cell 67, 2322333.E6 10.1016/J.Molchel.2017.06.007. PMC 5580496. PMID 2868965. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 41 Colo, Cassiope, Leggett DS, Wu Kilometer, Goldberg L., Finley D., June 2001. The axial channel of the proteasome core particle is gated by the RPT2 ATPase and controls both substrate entry and product release. Molecular cell 7, 6 114352 do 10.1016/S1097-2765, 01, 00274X. PMID 114301818. 42 Forster, Masters EI, Whitby FG, Robinson H, Hill CP, May 2005. The 1.9 A structure of a Proteasome 11S activator complex and implications for proteasome pan/par 700 interactions. Molecular cell 18, 558999 do 10.1000.j.molchel.2005.04.016 PMID 159169665 43 Witt S, Quan Yard, Sharon M, Feldera K, Butler M, Robinson CV, Baumeister W, Chap BK, July 2006. Proteasome assembly triggers a switch required for active site maturation. Structure 14, 7. 1179-88. Do 10.1016/j. Street 2006.05.019. PMID 168438999. 44 Kruger E, Cloacel PM, Enenkel C, 2001. 20 S Proteasome biogenesis. Biochimie. 83, 34289. 93 do 10.1016/S0300-9084, 01, 01241X. PMID 112954888. 45 Murata S, Yashirada H, Tanaka K, February 2009. Molecular mechanisms of proteasome. Assembly. Nature Reviews Molecular Cell Biology. 10, 21041152, 10.1038/NRM 2630. PMID 191652213. S2 SID 21263837. 46 Sakata E, Stengel F, Fukunaga K, Ju M, CKY, Forster F, Baumeister W, Tanaka K, Robinson CV. June 2011. The catalytic activity of UP6 enhances maturation of the proteasomal regulatory particle. Molecular cell. 42, 5637649 do 10.1016/j.molchel.2011.04.021. PMID 216588604. 47 Fukunaga K, Kudo T, Toyo, Tanaka K, CKY, June 2010. Dissection of the assembly pathway of the proteasome lid in Saccharomyces cerevisiae. Biochemical and biophysical research. Communications. 396, 4 1048 1053 do 10.1016/j.bbRc.2010.05.061. PMID 20471955. 48 Haas L, Worms JV, Hirschkow, Rose IA, March 1982. Ubiquitin activating enzyme. Mechanism and role in protein ubiquitin conjugation. The Journal of Biological Chemistry. 257, 2543 8. Do 10.1016/S0021-9258, 18, 34958-5. PMID 6277905. 49 Thrower JS, Hoffman L, Rieksteiner M, Picard cm, January 2000. Recognition of the polyubiquitin proteolytic signal. The EMBO Journal. 19, 194-102 do 10.1093/embodge/19.1.94 PMC 1171781 PMID 106198848 50 Recep, Daskalchukte, Banks TW, Louis, Cote Leasage J, Hellman H, Estelle M, Summerster. Crosby WL, June 2003. Protein interaction analysis of SCF ubiquitin E3 ligase subunits from Arabidopsis. The Plant Journal. 34, 675367 do. 10.1045-J.1365-313x.2003.01768.x PMID 127956996 51 Elsasser S, Finley D, August 2005. Delivery of ubiquitinated substrates to protein unfolding machines. Nature Cell Biology. 7, 87429 do. 10.1038/NCB0805-742. PMID 16056265. S2 SID 21069699. 52 Seydanandama, Bailey M, Ewan R, Lee J, Neelis S, October 2012. The ubiquitin proteasome system. Central modifier of plant signaling. The new phytologist. 196, 11328 do. 10.1111/J.1469. 8137.2012.04266.x. PMID 2289732. 53 Sharp PM, Lee W. H., 1987. Ubiquitin genes as a paradigm of concerted evolution of tandem repeats. Journal of Molecular Evolution. 25, 15864. Bibcode, 1987 Jmol 25, 58s. Do. 10.1007/BF02100041. PMID 3041010. S2 SID 7929162. 54 Picard cm, Fishman D., December 2004. Polyubiquitin chains, polymeric protein signals. Current. Opinion in Chemical Biology. 8, 661016 do. 10.1016/J.Kptar.2004.09.009. PMID 1555644. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 55 Zhu P, Zhuang DM, Seifried NT, Cheng D, Zai Y, Robert J, Rush J, Hochstrasser M, Finley D, Peng J. April 2009. Quantitative proteomics reveals the function of unconventional ubiquitin chains in proteasomal degradation. Cell 137, 113345 do. 10.1016/J.Cell.2009.01.041. PMC 2668214. PMID 193451992. 56 AB Picard cm, November 2000. Ubiquitin in chains. Trends in Biochemical Sciences. 25, 11544. 8 do. 10.1016/S0968-0004 00016881-9. PMID 11084366. 57 AB Zhang S, Zhu S, Yun D, Zhao L, Finley D, Wu Z, Mao Y, April 2022. USP 14 Regulated Illustrative. The Human Proteasome by Time Resolved Creo M. Nature. 605, 7910 5675574. Bibcode 2022 nature.605567Z. Do 10.1038/S41586-022-04671-8. PMC 91171149. PMID 35477760. 58 GQ, Waney G, Wang QE, Elmadi M, Snap KRM, Waney AA, July 2005. Dubiquitination by Proteasome is coordinated with substrate translocation for proteolysis in vivo. Experimental cell research 307, 243651 do 10.1016/j.yexca.2005.03.031. PMID 1595064. 59 Wenzel T, Baumeister W, March 1995. Conformational constraints in protein degradation by the 20s. Proteasome. Nature Structural Biology. 2, 3199204 do 10.1038/NSB0395-199. PMID 77737888. S2 SID 415999619. 60 Inob T, Fishbane S, Prakash S, Matuschko, March 2011. Defining the geometry of the two-component proteasome digron. Nature Chemical Biology. 7, 31617. Do 10.1038/Mchembio.521. PMC 31290032. PMID 21278740. 61 Van der Leer, Lang B, Cruz K, Xpona J, Sanchez de Groot N, Huinen Ma, Matuschko. Fuchsrieta M, Barbu mm, September 2014. Intrinsically disordered segments affect protein half-life in the cell and during evolution. Cell Reports. 8, 6 1832-44 do 10.1016/j.cellrip.2014.07.055. PMC 4358326. PMID 25220455. 62 Smith DM, Beeneralgen, Goldberger, October 2006. Proteasomes and their associated ATPases, a destructive combination. Journal of Structural Biology. 156, 172-83. Do 10.1016/j.jsb.2006.04.012. PMID 169194755. 63 Hoytma, Zik J, Takeki J, Chang M, Gavez C, Coffin OP, April 2006. Glycinylalanine repeats. Impair proper substrate unfolding by the proteasome. The EMBO Journal. 25, 8 1729. Do 10.1038/sj.embodge.7601058. PMC 1440830. PMID 166016992. 64 Zhang M, Coffin P., March 2004. Repeat sequence of Epstein-Barr virus encoded nuclear antigen. One protein interrupts proteasome substrate processing. The Journal of Biological Chemistry. 279, 10. 8635-41. Do. 10.1074/jbc.m3104492000. PMID 146888254. 65 C. Muller E., A. Lupus O., Stock D., Low J., Huber R., Baumeister W., April 1995. Proteasome from Thermoplasma acidophilum, a threonine protease. Science. 268, 5210 57982. Bibcode, 1995 SCI, 268579S. Do. 10.1126/science.77251007. PMID 7725107. 66 Kuzo, Tanaka K., Goldberg L., 1996. Structure and functions of the 20S and 26S proteasomes. Annual Review of Biochemistry. 65, 80147. Do. 10.1146/anav.by.65.0701960040101. PMID 881196. 67 Grohl M., Ditzel L., Low J., Stock D., Bochtler M., Bartunik H.D., Huber R., April 1997. Structure of 20S. Proteasome from yeast at 2.4 resolution. Nature. 386, 6624 46371. Bibcode, 1997 Nature.386463G. Do. 10.1038/386463A0. PMID 9087403. S2 SID 4261663. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 68 Dick TP, Nasbaum K, Degem, Heinmeier W, Grohl M, Schil M, Kielholz W, Stephen Orvik S, Wolf. DH, Huber R, Ramanzi HG, Schild H, October 1998. Contribution of proteasomal beta subunits to the cleavage of peptide substrates analyzed with yeast mutants. The Journal of Biological Chemistry. 273, 42563746 do. 10.1074/jbc.273.40.25637. PMID 9748229. 69 Vokes D, Zwicky P, Baumeister W, 1999. The 26S proteasome, a molecular machine designed for controlled proteolysis. Annual Review of Biochemistry. 68, 1015 Do. 10.1146/anav.biochem.68.1.1015. PMID 10872471. 70 AB Rape M, Gent Chess, May 2002. Taking a Bite, Proteasomal Protein Processing. Nature Cell Biology. 4, 5E1136 do. 10.1038/NCB0502E113. PMID 11988749. S2 SID 7126477. 71 Rape M. Gent Chess, November 2004. Productive rupture, activation of transcription factors by proteasomal processing. Biochimica et Biophysica Acta, BBA, Molecular Cell Research. 1695, 1. 320913. Do. 10.1016/j.bamker.2004.09.022. PMID 15571816. 72 Asher G, Royven N, Shawl Y, August 2006. 20 S Proteasomes and protein degradation by default. Bioessays. 28, 88449 do. 10.1002/bis.20447. PMID 169273316. 73 Zhang M, Picard cm, Coffin OP, April 2003. Determinants of proteasome recognition of ornithine. Decarboxylase, a ubiquitin-independent substrate. The EMBO Journal. 22, 1488-96. Do. 10.1093/embodge/cdg158. PMC 152902. PMID 126600156. 74 Asher G, Shawl Y, August 2005. 53 P Proteasomal degradation, polyubiquitination is not the whole story. Cell cycle. 4, 8 1015 8 do. 10.4161/cc.4.8.1900. PMID 16082197. 75 AB Sringapura, Grun T, Milhous J, Davies KJ, January 2003. Ubiquitin conjugation is not required for the degradation of oxidized proteins by proteasome. The Journal of Biological Chemistry. 278, 13118 do. 10.1004/jbc.m2062792000. PMID 12401807. 76 ABC Gil C, Goede, Schloet Elberg C, Priesner A, Cloacel PM, Gobel UB, Frummel C, March 2003. A comprehensive view on proteasomal sequences, implications for the evolution of the proteasome. Journal of Molecular Biology. 326, 1437-48 do. 10.1016/S0022-2836, 02, 01470-5. PMID 1259526. 77 Bochtela M, Ditzel L, Grohl M, Hartmann C, Huber R, 1999. The Proteasome. Annual Review of Biophysics and Biomolecular Structure. 28, 1295317 do. 10.1001/anav.biophys.28.1.295. PMID 104108004. 78 Chesnel F, Basil F, Pascalo, Cubic JZ, August 2006. Cyclin B dissociation from CDK1 proceeds. Its degradation upon MPF inactivation in mitotic extracts of Xenopus laevis embryos. Cell cycle. 5. 15 1687-98 do. 10.4161/cc.5.15.3123. PMID 169212258. 79 Brito The Reader CL, June 2006. Mitotic checkpoint slippage in humans occurs via cyclin B destruction in the presence of an active checkpoint. Current Biology 16, 12 1194 200. Do 10.1016/j.cub.2006.04.043. PMC 2749311. PMID 167820009. 80 Haven CG, Hoa, Yoshoka N, Dowdy SF, June 2006. Regulation of late G1/S phase transition and APCCDH1 by reactive oxygen species. Molecular and Cellular Biology 26, 12 470111. Do 10.1111/MCB.00303-06. PMC 1489138. PMID 1673833. 81 Bashir T, Doralo NV, Amador V, Gardavakaro D, Pagano M, March 2004. Control of the SCF, SKP2CKS1, ubiquitin ligase by the APC/C, CDH1, ubiquitin ligase. Nature 428, 6979 193. Do 10.1038/Nature 02330. PMID 150140502. S2 SID 440191. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 82 Higashitsuyi H, Lu Y, Maya RJ, Fujita J, October 2005. The oncoprotein gankirin negatively regulates both 53P and RB by enhancing proteasomal degradation. Cell cycle 4, 10.1335/7. Do 10.4161/cc.4.10.2107. PMID 161775771. 83 Tarason Reiser, Gabriel, Hertig, Frederick, Bray, Sean, Hafner, Annie. Harker Kirschneck, Lena, Foll. Peter, Davis, Colin, Papert Siamu, Dimitra, Mutov Kiev, Dulyan R. Fan, Catherine, Menigello, Letitia. Arashio Pulskin, Andre, Day, Gortame, Cully, Sean, Kilkenny, Mary, Souza, Diorge P. Pellegrini. Luca, De Bruin, Robertusa M. Henriks, Ricardo, Snijdes, Ambrosius P. Ari, Anella, Linders, Anne. Kristin, Robinson, Nicholas P. Baum, Buzz, 7 August 2020. The proteasome controls ISCAT3 mediated cell division in an archaeon. Science 369, 6504 as 2532. Do 10.1126/science.ars 2532. PMC 71160001. PMID 3276438. 84 Dermasiri S, Estelle M, 2002. The role of regulated protein degradation in auxin response. Plant molecular biology 49, 344019 do, 10.1023/101520301308. PMID 12036263. S2 SID 76693886. 85 Wijas D, Benkova E, Jagger K. Schleretha, Harman T, Keentz M, Wilmuth J.C., Reed J.W., Georgians. G. May 2005. Developmental specificity of AUGs in response by pairs of ARF and auxiliary/IAA transcriptional regulators. The EMBO Journal. 24, 10/18/74-85 do, 10.1038/sj.emboj.76000659. PMC 11425992. PMID 15889151. 86 Haasa L, Babashina O, Williams B, Schwartz L M, April 1995. Coordinated induction of the ubiquitin conjugation pathway accompanies the developmentally programmed death of insect skeletal muscle. The Journal of Biological Chemistry. 270, 16 9407 12 do, 10.1034/JBC.270.16.9407. PMID 772185. 87 Schwartz L M, Meyer, Koss L, Engelstein M, Meyer C, October 1990. Activation of polyubiquitin gene expression during developmentally programmed cell death. Neuron. 5, 44119. Do. 10.1016/0896-6273, 90, 90080Y. PMID 21697771. S2 SID 338297949. 88 Low P, Bussell K, Dawson SP, Billette Ma, Meyer RJ, Reynolds SE, January 1997. Expression of A. 26S Proteasome AT passe subunit, MS73, in muscles that undergo developmentally programmed cell death, and its control by ecdystroid hormones in the insect Manduca sexta. Feb's letters. 400, 3. 3459. Do. 10.1016/S0014-5793, 96, 01413-5. PMID 90092228. S2 SID 10873052. 89 Pitzer F, Dantezo, Fuchs T, Baumeister W, Amsterdam, September 1996. Removal of proteasomes from the nucleus and their accumulation in apoptotic blebs during programmed cell death. Feb's letters. 394, 14750 do, 10.1016/0014-5793, 96, 00920-9. PMID 892595. S2 SID 29256092. 90 AB Adams J, Palomella VJ, Sosville EA, Johnson J, Distria, Lazarus DD, Mass J, Pyan CS. Prakash S, Elliott PJ, June 1999. Proteasome inhibitors, a novel class of potent and effective antitumor agents. Cancer Research. 59, 11 2615 22. PMID 10363983. 91 Orlowski RZ, April 1999. The role of the ubiquitin proteasome pathway in apoptosis, cell death, and differentiation. 6, 430313 do 10.1038/sj.cdd.4400505. PMID 10381632. 92 Garrido C, Brunet M, Didelit C, Zermati Y, Schmidt E, Kroimer G, November 2006. Heat shock. Proteins 27 and 70, anti-apoptotic proteins with tumorigenic properties. Cell cycle. 5, 22 2592 601. Do 10.4161/cc.5.22.3448. PMID 171062661. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 93 Parkshire, Bolander N, Isle F, Kostover Z, Takey J, Coffin OP, Wolf DH, January 2007. The cytoplasmic HSP70 chaperone machinery subjects misfolded and endoplasmic reticulum import incompetent proteins to degradation via the ubiquitin proteasome system. Molecular biology of the cell. 18, 115365 do 10.1091/mbc.e06-04-0338. PMC 1751312. PMID 170655559. 94 DIQ, Chien SB, Li HH, McDonough H, Borchers C, Huang D, Takayama S, Younger JM, Ren HY, CYR. DM, Patterson C, November 2005. Regulation of the cytoplasmic quality control protein degradation pathway by BAG2. The Journal of Biological Chemistry. 280, 46 3867 381. Do 10.1074/jbc.m5079862000. PMID 161698550. 95 Bader N, Grun T, 2006. Protein oxidation and proteolysis. Biological chemistry. 387, 1011. 1351 5. Do 10.1515/bc.2006.169. PMID 17081106. S2 SID 30385354. 96 Davies KJ, 2003. Degradation of oxidized proteins by the 20S proteasome. Biochemie. 83, 34. 30110. Do 10.1016/S0300-9084, 01, 01250-0. PMID 11295490. 97 AB Lehrman NL, September 2009. The ubiquitin proteasome system in neuropathology. Acta Neuropathologica. 118, 332947 do 10.1074/00401-009-0560X. PMC 2716447. PMID 195978929. 98 McNaught KS, Jackson T, Chno Baptista, Capistino, Olanal CW, May 2006. Proteasomal dysfunction in sporadic Parkinson's disease. Neurology. 66, Tensor 4S 3749. Do. 10.1200/01.WNL.000221745.58886.2e. PMID 167172151. 99 Sharma N, Brandis K.A., Harira S.K., Johnson B., Vaidya T., Shrestha R., Deberman S.K., 2006. Alpha-synuclein budding yeast model, toxicity enhanced by impaired proteasome and oxidative stress. Journal of Molecular Neuroscience. 28, 216178 do. 10.1030/JMN28:2:161. PMID 166795556. S2 SID 27762513. 100 Murata S, Sasaki K, Kishimoto T, Niwa S, Hayashi H, Takahama Y, Tanaka K, June 2007. Regulation of CD8 plus T cell development by thymus-specific proteasomes. Science. 316, 5829 1349. 53 Bib code, 2007 SCI, 316.1349 Do. 10.1126/science.11419115. PMID 175409004. S2 SID 37185716. 101 Cassiope, Hilton C, Kiselev, Rock KL, Goldberg L, May 2001. 26 S proteasomes and immunoproteasomes produce mainly N-extended versions of an antigenic peptide. The EMBO Journal. 20, 10.235766 do. 10.1093/emboj/20.10.2357. PMC 125470. PMID 11350924. 102 Mallory DL, McEwen WA, Biggood SR, Towers GJ, Johnson cm, James LC, November 2010. Antibodies mediate intracellular immunity through tripartite motif containing 21, Trim 21. Proceedings of the National Academy of Sciences of the United States of America. 107, 46 19985. 19990 Bib code, 2010 as 107199885M. Do. 10.1073/NAS.1014074107. PMC 2993423. PMID 21045130. 103 Fentini G, Standair RF, Lane WS, Choi S, Corey EJ, Schreiber SL, May 1995. Inhibition of proteasome activities and subunit-specific aminoterminal threonine modification by lactacystin. Science. 268, 5211 72631. Bib code, 1995 SCI, 268726F. Do. 10.1126/science.77323882. PMID 77323882. S2 SID 37779687. 104 United States Food and Drug Administration press release archived 19 February 2007 at the Wayback Machine 13 May 2003. Access date 29 December 2006. See also FDA Velcade information. Page. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 105 Fisher I, Bernstein Sh, Kyle B.S., Julbigovic B., Robertson M.J., DeVos S., Ebner E., Krishnan O. Leonard J.P., Loniel S., Statmore E.A., O'Connor O.A., Shi H., Borel L., Boyer, October 2006. Multicenter Phase 2 study of bortezomib in patients with relapsed or refractory mantle cell lymphoma. Journal of Clinical Oncology. 34867 do. 10.1200/JCO.2006.07.9665. PMID 17001068. 106 Jacob C., Ijira K., Libis P., Turkman S., Zavsky I., Kukulkan U., Haider U., Kaiser M., Fleisner C. Stott J., Kleberg L., Feist E., Burmister Gr., Cloacel P.M., Cesar O., March 2007. Circulating. Proteasome levels are an independent prognostic factor for survival in multiple myeloma. Blood. 109. 5 2105 do. 10.1182/blood 2006-04-016360. PMID 17095627. 107 Shah Sat, Potter M.W., Mcdade T.P., Rachir D.R., Pereginira, Elliott P.J., Adams J., Caller E.M.P., 2001. 26S Proteasome inhibition induces apoptosis and limits growth of human pancreatic cancer. Journal of Cellular Biochemistry. 82, 111022 do. 10.1022/JCB.1150. PMID 11400168. S2 SID 21223980. 108 Noroki St, Sweenagoch B, Takamori R, McConkey DJ, January 2004. The proteasome inhibitor bortezomib enhances the activity of docetaxel in orthotopic human pancreatic tumor xenografts. Molecular cancer therapeutics. 3, 15970 do. 10.1585/1165/7163.59.3.1. PMID 14749476. S2 SID 38429730. 109 Skenkine D., June 2002. Proteasome inhibitors in the treatment of B-cell malignancies. Clinical lymphoma. 3, 14955 do. 10.3816/CLM.2002.N.011. PMID 12141956. 110 O'Connor O.A., Wright J., Moskowitz C., Muzzy J., McGregor Cortelli B., Stubblefield M., Strauss D. Portlock C., Hamlin P., Choi E., Dumetrescu O., Iseltine D., Trihu E., Adams J., Skenkine D., Zelanets. A.D., February 2005. Phase 2 clinical experience with the novel proteasome inhibitor bortezomib in patients with indolent non-Hodgkin's lymphoma and mantle cell lymphoma. Journal of Clinical Oncology. 23, 467684 do. 10.1200/JCO.2005.02.050. PMID 156136999. 111 Messinger year, Gainan PS, Spistoa, Van der Giesen J, Ekrothi, Malvar J, Bostrom BC, July 2012. Bortezomib with chemotherapy is highly active in advanced B precursor acute lymphoblastic leukemia. Therapeutic advances in childhood leukemia and lymphoma, tackle, study. Blood. 120, 2. 28590. Do. 10.1182/blood 2012-04-418640. PMID 226539776. 112 Lambrugii, Papadimitriou L, Chrysos GP, Vlahopoulos Sat, April 2012. Glucocorticoid and proteasome inhibitor impact on the leukemic lymphoblast, multiple, diverse signals converging on a few key downstream regulators. Molecular and cellular endocrinology. 351, 214251. Do. 10.1016/J.Mas.2012.01.003. PMID 22273806. S2 SID 28749125. 113 Schmidt G, Holhutter HG, Bogio M, Caries N, Grohl M, De Gaiulia, M. Chess, Groetrup M. December 1999. How an inhibitor of the HIV-I protease modulates proteasome activity. The Journal of Biological Chemistry. 274, 53573440 10.1074/JBC.274.50.35734. PMID 1058554. 114 Lauron N, De Bois S, Guillermo J.S., Christophe C., Zinnia, Chwalt H., Andre P., Lotti O.V., Peschansky M., February 2004. Effects of the proteasome inhibitor ritonavir on glioma growth in vitro and in vivo. Molecular Cancer Therapeutics. 3, 212936 do. 10.1558/1535-7163.129.3.2. PMID 149854553. S2 SID 244233806. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 115 Zollner TM, Podder M, Pyan C, Elliott PJ, Kaufman R, Bonk WH, March 2002. Proteasome inhibition reduces superantigen-mediated T-cell activation and the severity of psoriasis in a situ model. The Journal of Clinical Investigation. 109, 56719 do. 10.1172/JCI12736. PMC 150886. PMID 1187745. 116 Elliott PJ, Pyan CS, McCormick Ta, Chapman ID, Adams J, August 1999. Proteasome inhibition, a novel mechanism to combat asthma. The Journal of Allergy and Clinical Immunology. 104, 2 PT1. 294300. Do 10.1016/S0091-6749 99 70369-6. PMID 104524747. 117 Verdos M. Floria by Menendez Benito v. Maynard C.J., Witt M.D., Van der Linden W.A., Van den Nieuwendigem, Hoffman T., Burkers C.R., Van Leeuwen F.W., Gruthuis Ta, Leeuwenberg Ma, Ovara H., Nephews J.J., Philippof D.V., Van der Merel G.A., Dantuma N.P., Overcleft H.S., November 2006. A. Fluorescent broad-spectrum proteasome inhibitor for labeling proteasomes in vitro and in vivo. Chemistry and Biology. 13, 11 1217 26 do. 10.1016/j.chembiol.2006.09.013 hdl 1887/65477. PMID 171140003. 118 Klieger G., Mayer T., June 2014. Perilous Journey, a tour of the ubiquitin proteasome system. Trends in Cell Biology. 24, 63529 do. 10.1016/j.tcb.2013.12.003. PMC 4037451. PMID 244570024. 119 Goldberg L., Stein R., Adams J., August 1995. New Insights into Proteasome Function, from Archaebacteria to Drug Development. Chemistry and Biology. 2, 85038 10.1016/1074. 5521, 95, 90182-5. PMID 9383453. 120 Sulistio Ya, Hees K., January 2015. The Ubiquitin Proteasome System and Molecular Chaperone. Deregulation in Alzheimer's Disease. Molecular Neurobiology. 53, 290531 do. 10.1007/S12035. 014-9063-4. PMID 255614338. S2 SID 14103-185. 121 Ortega Z., Lucas J.J., 2014. Ubiquitin Proteasome System Involvement in Huntington's Disease. Frontiers in Molecular Neuroscience. 7, 77. Do. 10.3034/fmol.2014.00077. PMC 4179678. PMID 253241717. 122 Sundry M., Robbins J., June 2014. Proteotoxicity, an underappreciated pathology in cardiac disease. Journal of Molecular and Cellular Cardiology. 71, 310. Do. 10.1016/j.yjmcc.2013.12.015. PMC 4011959. PMID 2438070. 123 Druzo, Teicht Mayer H., December 2014. Targeting the Ubiquitin Proteasome System in Heart Disease, the Basis for New Therapeutic Strategies. antioxidants and redox signaling. 21, 17 2322 43. Do. 10.1000/rs.2013.5823. PMC 4241867. PMID 251336888. 124 Wang ZV, Hill J.A., February 2015. Protein Quality Control and Metabolism, Bidirectional Control in the Heart. Cell Metabolism. 21, 221526 do. 10.1000/j.cmd.2015.01.016. PMC 4317573. PMID 256511176. 125 AB M, Delace M, February 2000. The I-kappa-B kinase, IKK, and NF-kappa-B, key elements of pro-inflammatory signaling. Seminars in Immunology. 12, 185-98. Do. 10.1006/SMIM.2000.0210. PMID 107238001. 126 Termo Ma, Dak Hofnicker, Schumacher B., January 2015. Quality Control Mechanisms in Cellular and Systemic DNA Damage Responses. Aging Research Reviews. 23, PTA 311. Do. 10.1016/j.2014.12.009. PMC 4886828. PMID 2556047. 127 Chekler F., the Costa Ca., Ancalayo K., Chevalier N., Lopez Perez E., Marambord P., July 2000. Role of the proteasome in Alzheimer's disease. Biochimica et Biophysica Acta, BBA, molecular basis of disease. 1502, 11338 do. 10.1016/S0925-4439, 000039-9. PMID 1089948. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 128 AB Chung KK, Dawson VL, Dawson TM, November 2001. The role of the ubiquitin proteasomal pathway in Parkinson's disease and other neurodegenerative disorders. Trends in Neurosciences. 24. 11 Supple S714 do. 10.1016/S0166-2236, 00019998-6. PMID 118817848. S2 SID 22111658. 129 AB Ikeda K, Akiyama H, Airite, Uyeno H, Sachiya K, Kosei Ka K, July 2002. Morphometrical reappraisal of motor neuron system of Pick's disease and amyotrophic lateral sclerosis with dementia. Acta Neuropathologica. 104, 121 do. 10.1007/S00401-001-05113-5. PMID 120706660. S2 SID 22396490. 130 Manekar H, Kato T, Kurita K, Katajiri T, Shikama Y, Kujirai K, Kawanami T, Suzuki Y, Nihir K, Sasaki H. May 1992. Marked increase in cerebrospinal fluid ubiquitin in Creutzfeldt-Jakob disease. Neuroscience Letters. 139, 147 9 do. 10.1016/0304-3940, 92, 90854Z. PMID 1328965. S2 SID 28190967. 131 Matthews KD, Morsa, January 2003. Limb-girdle muscular dystrophy. Current Neurology and Neuroscience Reports. 3, 17885 do. 10.1077/119110-003-0042-9. PMID 125074116. S2 SID 5780576. 132 Meyer RJ, March 2003. From neurodegeneration to neurohomeostasis, the role of ubiquitin. Drug. News and Perspectives. 16, 21038 do. 10.1038.dnp.2003.16.2.82937. PMID 1279267. 133 Calise J. Powell S.R., February 2013. The Ubiquitin Proteasome System and Myocardial Ischemia. American Journal of Physiology. Heart and Circulatory Physiology. 304, 3H33749. Do. 10.1152/Archfert.00604.2012. PMC 3774499. PMID 232203331. 134 Pridmore J.M., Wang P., Davis F., Bartolone S., Westfall M.V., Dyke D.B., Pagani F., Powell S.R., Day S.M. March 2010. Ubiquitin proteasome dysfunction in human hypertrophic and dilated cardiomyopathies. Circulation. 121, 8997 1004 do. 10.1161/circulation/AHA.109.904557. PMC 2857348. PMID 201598828. 135 Powell S.R., July 2006. The Ubiquitin Proteasome System in Cardiac Physiology and Pathology. American Journal of Physiology. Heart and Circulatory Physiology. 291, 1H1H19. Do. 10.1152/Archfert.00062.2006. PMID 1650126. S2 SID 7073263. 136 Adams J., April 2003. Potential for proteasome inhibition in the treatment of cancer. Drug discovery. Today. 8, 730715 do. 10.1016/S1359-6446, 03, 02647-3. PMID 126545543. 137 Benneria Y., January 2002. Regulatory functions of ubiquitination in the immune system. Nature. Immunology. 3, 126 do. 10.1038/Ni0102-20. PMID 117534006. S2 SID 26973319. 138 Karatijara K, Kukulkan U, Rudolf P, Ruckert J.C., Dorna T, Burmester G.R., Cloacel P.M., Feisty. October 2002. Circulating proteasomes are markers of cell damage and immunologic activity in autoimmune diseases. The Journal of Rheumatology. 29/10/2045-52. PMID 123753110. Further reading, edit. Glickman M.H., Adir N., January 2004. The Proteasome and the Delicate Balance Between Destruction and Rescue. PLOS Biology. 2, 1E13 do. 10.1071/journal.bio.00200013. PMC 314468. PMID 1473789. The Yeast 26S Proteasome with list of subunits and pictures. Sechanova, September 2005. Early work on the ubiquitin proteasome system, an interview with Aaron Sechanova. Interview by CDD. Cell death and differentiation. 12/9/1167-77. Do. 10.1038/sj.cdd.44016991. PMID 160943993. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Hershkower, September 2005. Early work on the ubiquitin proteasome system, an interview with Avram Hershko. Interview by CDD. Cell death and differentiation. 12/9/1158-61. Do. 10.1038/sj.cdd.44017009. PMID 160943991. Rose I, September 2005. Early work on the ubiquitin proteasome system, an interview with Erwin Rose. Interview by CDD. Cell death and differentiation. 12/9/1162-6 do. 10.1038/sj.cdd.44017000. PMID 160943992. Kvek B, Dvorak Z, 2007. Targeting of nuclear factor Cap-Hab and proteasome by dithiocarbamate. Complexes with metals. Current pharmaceutical design. 13/33155-67. Do. 10.2014/1381612077821103990. PMID 179799756. Archived from the original on 29 July 2012. External links, edit. Proteasome Subunit Nomenclature Guide. 3D proteasome structures in the M-Data Bank, EMDB. Key points of proteasome archived 17 November 2020 at the Wayback Machine function. Categories: proteins, protein complexes, organelles, apoptosis. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.